When we talk about DNA replication in eukaryotic cells, there is a bit of a problem presented because these are linear chromosomes. And so if we take a closer look at what the actual problem is that we have with the tips of the chromosomes, as you can see here, we have this primer actually on this very end of the chromosome. And that would have been placed there by the primase enzyme. And recall that that primer is made of RNA. And that's not really what we want inside the DNA because we want just consistent, continuous DNA there. But it did serve a purpose. That primer provided the 3' end for DNA polymerase to extend. So once we get the DNA polymerase extended, as you see right here, in eukaryotic cells, it's actually going to be RNase H that comes in and removes that particular primer from the DNA because we don't want it there. And then once that is removed, this would be a 5' end. And our problem is now how to actually synthesize or make this extra DNA right here. Because if you'll recall, DNA polymerase cannot add to a 5' end. And that's why we had the primer to start with. So the question is, how do we actually get this extra piece synthesized, because we can't really back up any further and put the primer on. If we put a primer on there, we'd be in the exact same situation that we had before. So we're going to have yet another enzyme that's going to help with this process in particular. This enzyme's name is telomerase, and it's called telomerase, and ACE indicates it's an enzyme, but it's called telomerase because the very tips of chromosomes are called telomeres. So telomerase's job is going to be to extend the telomere, basically make it longer so that vital information, and that would be vital information in our DNA, is not lost during DNA replication. And that's really important because one of the big goals with DNA replication is to take all the content inside the cell and copy it exactly so that we will get these functioning cells at the end. Okay, so that's going to be telomerase's job. So let's take a little bit of a closer look at what that enzyme is actually going to be doing. So here we see a close-up look at the telomere, or the end of the chromosome, and telomerase itself is going to have sequence that is complementary to this very tip of the chromosome. And by complementary, we're going to have the matching base pairs, so the matching bases that can hydrogen bond with what we see right there. Also with telomerase, it is RNA sequence that it has. So since it's RNA sequence, we will see uracil as the base instead of thymine there. So I'm going to write this sequence in green because it is telomerase sequence, which is actually RNA sequence. And I'm going to add that on here. It's actually part of the telomerase enzyme. And so this is what we're going to end up with. Okay, so when we look at that, what's going to happen is that now we can actually extend the piece of DNA that's on the top. So if we look at that, we can have a DNA polymerase enzyme that actually comes in and starts adding the correct complementary bases here. Okay, so notice that my end just got a little bit longer. And this can happen again as well, and sometimes it does happen multiple times in a row. So if I was to erase this, that same sequence is going to be complementary to some sequence that's a little further down. So I don't know if you noticed or not, but this sequence right here is the same as this sequence right here, which means that that same telomerase enzyme can now come and actually add on right here and extend again if there's a need for that. And the sequence goes a little bit longer. And if we did that, we could have the DNA polymerase come in, and it again could come in here and actually extend that top piece a little bit more. So I'm going to write in here as though it was extended just a little bit more. So this is now my 3' end over here. Well, the telomerase enzyme goes away, and with it, so does its RNA piece, because that's just part of the enzyme itself. But the significant thing that has happened here is notice that the end of the chromosome is longer. And the benefit for this is that now we have a place to put another primer. Okay, so primase, that's its job. So primase is going to come in here and it's going to add its complementary primer. It's going to start at the very end right here. And I didn't specify the bases there, but it's going to add its primer in here. And then, so this would be my new 3' end. And once we have the new 3' end, then DNA polymerase can come in and it can add its sequence. So I'm going to go ahead and write that in, what DNA polymerase would be adding. It would be adding the correct base pairs. And then once it has done that, we would be relying on the ligase enzyme. So we will have ligase actually come in and make that final link, that final phosphodiester linkage there. So that's still going to happen. And then we are going to have, since this is a eukaryotic cell, RNase H will come and remove the primer. Okay, so let me just write that down here so that we do understand that we are going to get removal of that particular primer. So we do still have this area on the end that did not get actually copied. So this part right here, but that was extra sequence that the telomerase enzyme added. So the significant thing here is that the chromosome got a little longer because of the activity of telomerase. And so when the chromosome has that area that we can't replicate, that would just be extra DNA sequence. It's not going to be that vital sequence of the chromosome that actually tells the cell how to make something.